Good afternoon, and welcome to the American Floral Endowment's Grow Pro webinar series. I'm your moderator, Mike Wajasinga, and I'm a grower for Bell Nursery in Springfield, Ohio. I help oversee and manage the production of a wide variety of ornamentals for hundreds of stores across the country. Today's session is on sustainable disease management solutions for cut flower growers. On behalf of the endowment, I'm excited to be part of AFE's Grow Pro webinar series that features a new topic every month presented by an industry expert. The webinars are free to everyone thanks to the generous support of AFE's sponsors. This session is sponsored by Grower Talks and OHP. Grower Talks is a well-known greenhouse trade publication produced by Ball Publishing that brings readers the latest in growing and retailing information. OHP's mission is to provide high-quality pest control products and services to the greenhouse and nursery production industry in order to provide grower solutions. If you'd like to learn more about our sponsors, or if you're a supplier and interested in becoming a sponsor for a topic, you can find that information on AFE's website at endowment.org/growpro. Today's session was pre-recorded in English by Dr. Francesca Hand. After the presentation, Dr. Hand will join us for a Q&A. Feel free to submit your questions through the Q&A feature or chat at any time during the presentation. We'll answer as many as we can before the end of the hour. Unanswered questions may be answered through a separate email exchange. This session is being recorded and shared to AFE's YouTube account. Through the YouTube's accessibility features, you can access closed captions in other languages. To get us started, I'd like to share a bit about our expert speaker, Dr. Hand. She is an associate professor of plant pathology and state extension specialist for ornamental plant pathology at The Ohio State University. Previously, she received a PhD in plant pathology from the University of Florence, Italy, and she conducted postdoctoral research at the University of California, Davis. The purpose of her research and extension program at OSU is to investigate disease epidemiology, biology, and ecology of fungi, fungal, oomycete, and bacterial pathogens, from which she develops detection and control strategies. Dr. Hand, welcome, and thank you for presenting on today's topic, Sustainable Disease Management Solutions.
All right. Thank you, Mike, for the invitation to speak today during this webinar series. This is a great opportunity for me to share some of the work that we're conducting in my program at The Ohio State University. And most of the information that I will share today was developed and collected by my graduate student, Daniela Gutierrez-Yanes, who you will see in many of the slides, and was done in collaboration with Dr. Anna Testen from USDA ARS in Worcester, Ohio. So as you know, the floriculture industry is a big component of the US economy and contributes approximately $6.5 billion in annual national sales. Ohio, where I am located, is among the top 5 producers of floriculture products, and the small state contributes about $2 million to the total. Cut flowers represent a big part of the floriculture industry, and they contribute just short of about $400 million in annual national sales. And within the cut flowers is a special sector that has been experiencing tremendous growth in the last few years, and this is the specialty cut flower sector. Flower crops that belong to this category of the industry are flowers that don't ship well or have a fairly short post-harvest life, and for that reason are not imported and are mostly sold locally where they are produced. There's examples of crops here, but there's many, many different crops that fall into this group. And about 90% of them is sold on the wholesale market, and then the rest of it is sold either directly to florists or made available for U-Peak on roadside stands, and populates our beloved farmer markets, which really contribute to the development of local communities and local economy. And this growth of the sector is well reflected in the increase in membership that the Association of Specialty Cut Flower Growers has experienced in the last few years, where their membership has increased more than 100%. So cut flower production systems, including specialty cut flowers, are very diverse. And this doesn't just refer to the variety of crops that are grown within a farm at any given time, but also it refers to the diversity of production systems. For example, some crops are grown in crates, these containers. We're referring specifically to crops such as lilies. And in these containers, in these crates, we often have a combination of soilless media amended with compost or other organic and inorganic matter. It includes ground beds in high tunnels or open fields where the crops are grown directly in field soil. And then it includes raised beds, where again, often a mix of soilless substrates and soil-based substrates and compost and other amendments are used. Now, when crops are grown in these different production systems, whether they are ground beds in the field or inside in protected agriculture, often we use high tunnels, and we see an increase in number of growers that chooses to grow their crops in high tunnels, because these structures can provide an extension to the growing season, which is clearly desirable, but also can provide crops that have longer stems and bigger caliper of the stems and the blooms, which also are very desirable characteristics. Most of the time, we see that crops that are grown in the field are grown in fields that have been repurposed from field crop production. That, how often we see cornfields and soybean fields and wheat fields that have now been repurposed for the production of cut flowers. And of course, these can come with many challenges, because these field crops are very well known for being reservoirs of soilborne pathogens. So pathogens that naturally live in the soil and then can reach very high inoculum concentration in the soil, constituting a risk for whatever crops is going to be planted in these fields. So as I mentioned, production of cut flowers can use a variety of substrates. These include soilless substrates and soil-based substrates, and there's risks that come with both of these groups of substrates and a combination of them. We can have soilless substrates that are usually free of pathogens, but then often growers, to cut down production costs, may decide to recycle these substrates without proper disinfestation, or mix these substrates with field soil that I just mentioned is always a risk for introduction of soilborne pathogens. And these can often lead to very bad outcomes, such as the one that you see here in this cut flower farm, where many of the beds that are included in this high tunnel were completely devastated by soilborne pathogens. In this case, specifically, was Rhizoctonia diseases and Pythium root rot. So based on what outlined in the last few slides, I sought to define some program goals to align with industry needs. And because the first step in any disease management plan should be the correct identification of the causal agents of disease, I planned on conducting a survey of cut flower producers, specialty cut flower producers specifically, to identify what were the major disease problems that they faced. And this work was funded locally by the Ohio Department of Agriculture initially. And the results of this survey fed into the development of informational material that was then funded by the American Floral Endowment in combination with the Association of Specialty Cut Flower Growers, where the goal is to develop a disease guide where information on the pathogens causing disease, images of symptoms and signs and recommendations for management are going to be made available to the industry. Within these goals was also that of exploring the use of alternative disease management options for controlling soil-borne pathogens that don't rely on the use of pesticides. And this is both in the lens of environmental sustainability, but also because many of the specialty cut flower growers, especially the small growers, do not really use much pesticides. And so having tools available that are effective and have a broad impact can be extremely helpful. So within these goals, we conducted a sampling of cut flower farms. And in the last year, we have looked at approximately 100 different samples. The majority of them came from outdoor production. So we're talking about raised beds or ground beds in field production. From protected agriculture, and then about 40% from outdoor production. And then within these two production systems, more than 3/4 of the samples came from in-ground beds, again, highlighting the importance of soil as a potential introduction of soil-borne pathogens. When we received the samples, we proceeded to observe the symptoms and the signs present on the samples. We took very good pictures that we can then include in the disease guide that will be available in the next year or so. And then we conducted a variety of tests, from basic tests such as immunostrip assays for detection of viral diseases and oomycete diseases, to culturing of pathogens, so extraction of pathogens from the plant, identification of the pathogens using morphological features, all the way to analysis of their DNA to get a better idea of what genera and species of these pathogens we were dealing with. Here is kind of a summary of that investigation. So in about 60% of the samples that we processed, we were able to identify a pathogen as the culprit of the symptoms. In fewer instances, we found insect-related damage or environmental-related damage, and in about 25 to 27% of the cases, we were unable to determine what was the cause of the issue for a variety of reasons. But of those 62% of samples that showed that a pathogen was causing the problem, more than 75% was attributed to either a fungus or an oomycete. And oomycetes are water molds. They are pathogens such as Pythium, Phytophthora, or even downy mildew that are very aggressive pathogens that are very much present in field soil and can contribute greatly to crop losses in cut flower production. Here are some examples of the symptoms that we observed. As you can see here, we have the thinning of the stems just below the soil line where the root is attached. And that is a characteristic symptom of Pythium root rot. We saw a lot of sloughing of the roots as a consequence of the pathogen killing that root system. We had a lot of cases in which we found vascular diseases such as Fusarium wilt. Fusarium is a very common pathogen in specific crops, mostly in Lisianthus. We had a lot of Lisianthus samples that had Fusarium wilt, and you can see the discoloration here inside the vascular tissue that is exposed by peeling off that external layer of the plant. And in advanced stages, you can see the mycelium, which is the body of the fungus that is growing out of that vascular tissue, causing the plant to wilt and extensive loss. In some other instances, we found other soil-borne pathogens, such as sclerotinia, that was colonizing, again, the stem by the head of by the flower head, causing the plant to lodge and the plant to collapse. A lot of foliar diseases were also found. Many of them are soil-borne in origin. For example, downy mildew here. This was very common on our Matiola stalks. We found evidence of rust on crops such as snapdragon, and then some bacterial diseases such as Xanthomonas on sunflower. And then another example is of anthracnose. We found a lot of anthracnose on peony, where the pathogen infects all kinds of tissue in that plant, the flower, the leaves, the stems, causing these necrotic lesions that misshape the bloom, making the plant unsellable. So using all these information and the images that we collected, we are in the process, as I said, of developing different educational resources. One of them will be this disease guide that is in the process of being developed, and some of them fed into the development of quick cheat sheets where some images of symptoms and signs are provided along with very brief information about what is causing the problem, how that problem can spread within the production, what are conditions that favor disease developments, and ultimately what we can do about that. And again, there's a variety of different educational material that we will develop with all these images and information on the pathogen that we are collecting. We'll still have a year of funding for this project, so we'll continue to do that throughout next year. So it became very clear, and we kind of knew these already, but it became very clear that soil-borne pathogens are the main culprit of problems in specialty cut flower production. And so we decided to look into options about soil-borne disease management. And one of the key factors in managing diseases caused by soil-borne pathogens is the reduction of the inoculum level in the soil. And this has historically been done with chemical fumigation, using chemical fumigants such as methyl bromide, which has now been banned and is no longer available because it has such a negative impact on soil health. And with methyl bromide, many other fungicides or fumigants have been either phased out or heavily restricted. However, on the other hand, many cut flower producers are choosing to adopt more sustainable practices, as I mentioned earlier, and some of them include soil solarization and soil steaming. In one instance, in the soil solarization, the technique uses the sun heat to kill the microorganisms that are in the soil. In the other case, we have steam that pretty much has the same effect. Now, soil solarization can be effective, but it is an option that is not feasible in temperate climates, such as the ones in the Midwestern and Northeastern United States. And soil steaming is a relatively new technique applied to the production of cut flowers, and there's a lot of uncertainties about the duration of the treatment, the depth at which the treatment should be reaching, and also how long would the benefit of the steaming last after the treatment. So in addition to these three options for soil-borne disease management is a relatively new technique known as anaerobic soil disinfestation, or ASD. This is a technique that consists of 3 steps. In the first step, we incorporate a easily decomposable carbon source into the soil. Then we irrigate the soil to saturation. And then finally, we cover the soil with a turf and create anaerobic conditions and maintain these anaerobic conditions for a variable amount of time. And these variability in time of the treatment depends on the type of soil, the temperature that is reached. So it's a technique that needs to be adapted based on the environment in which it is conducted. During these weeks of incubation, in which the anaerobic conditions are created, the composition of the microbes in the soil shifts from mainly aerobic pathogens to anaerobic pathogens. And these anaerobic anaerobic microorganisms thrive in this environment that now has very little oxygen. And this anaerobic condition promotes their growth and promotes the development of toxic compounds produced by these pathogens, by these microorganisms that kill the pathogen, and also allow them to be more competitive for food sources. And all of the effect of these factors combined leads to suppression of soilborne pathogens. So the use of ASD has been, in the last few years, has been applied to a lot of different cropping systems, and these include mainly vegetables and strawberries. But there are just a few studies that have actually looked at using ASD for the production of ornamental crops. And specifically for cut flowers, for flower crops, there are only 3 studies that have been done, one in Florida, one in Argentina, and one in China, where the target were either the root knot nematode in Florida or Fusarium oxysporum in the other 2 instances. These 3 studies have used different carbon sources. As you can see, it was either molasses or wheat bran or bagasse, and the incubation period was very similar. The temperatures that were reached were very similar. And you can note that all these environments are in tropical or subtropical regions. And so we have very high temperatures, environmental conditions that are very, very different from what we see in the temperate climates. So when this technique is explored for application in temperate climates, it must be tested for its suitability. And so in Ohio, ASD has been extensively tested in vegetable production systems in either field-grown vegetables or high tunnel grown vegetables. And this was done by my collaborator, Dr. Anna Testen, and at that time, her graduate advisor, Dr. Sally Miller. And Dr. Testen and Dr. Miller looked at a lot of different carbon sources that were readily available in Ohio, which is one of the things that is important to consider when recommending the use of this technique to growers. And they looked at an incubation period of about 4 weeks at 25 Celsius. And they identified that if this temperature could be maintained for a period of 4 weeks, that ASD was a suitable option. So based on this preliminary information, we decided to develop a proof of concept study to see if ASD could be a suitable option for the production of specialty cut flowers in the temperate regions of the United States. So we selected 2 different pathosystems. We used Rhizoctonia solani, which is a fungus that causes stem rot in many flower crops, and we paired that with zinnia as our plant system. And then we looked at Phytophthora root rot and crown rot with Phytophthora drexlerii on Gerbera daisy. So this is my graduate student up here that did the work, and what she did is she grew the pathogen in vermiculite, so a completely inert substrate that was fully infested with the pathogen. And then she mixed these vermiculite infested with the pathogen with either soilless substrates or soil-based substrates. The reason why we decided to evaluate the technique for both soilless and soil-based substrates is because of what I mentioned initially, the fact that the industry actually heavily uses either or a combination of both of these substrates. So the soilless substrate that we used was Promix BX, And then the soil-based substrate was a locally produced soil that is used in raised bed production called Jones super soil that contains a high amount of comb-till mixed with sand. So both of these substrates that were mixed with the pathogen were now mixed again with either wheat bran, tomato pomace, or soybean meal as our three different carbon sources. And we picked these three carbon sources because of their availability locally in Ohio, but also because they were previously tested in vegetable system. Specifically, wheat bran is considered the gold standard carbon source, so it's the one that we wanted to compare everything else against. Soybean meal is extremely available in Ohio, and tomato pomace had been identified in laboratory studies as a potential good carbon source for ASD. And we compared these three carbon sources with a control where there was only the soil with the pathogen, but without the additional carbon amendment. All these treatments were placed inside Ziploc bags, and then water was added to the bags to saturate the soil to mimic those saturation conditions that we would achieve in the field. And then we sealed the bag and relieved the air, put it inside another bag to make sure that it would maintain those anaerobic conditions for the time being. We also included a control where we didn't create anaerobic conditions. That was our aerobic control that was treated exactly the same, except that the bags weren't sealed. Now you will notice that there is a little tube that was added to each of these bags, and this tube was coated with iron oxide paint. And iron oxide paint was our measure of how much of the anaerobic conditions were reached inside the bag. If anaerobic conditions were reached, we would expect the iron oxide paint to be completely dissolved. And then in case the anaerobic conditions weren't reached, then the iron paint would still be coating the PVC tubes. We placed these bags inside an incubator for 4 weeks at 25 Celsius. And then at the end of that incubation process, we opened the bags and we transferred the content of the bags inside pots and planted a plug of either zinnia or Gerbera daisy inside each pot. Placed the pots in a greenhouse and observed development of disease for 3 to 5 weeks, depending on the system. So we measured different variables throughout this experiment. As I mentioned, we looked at how much anaerobic conditions were reached in the different treatments. And here you have an example of the oxide paint that did not disappear from the PVC tubes to this group down here where we had almost complete loss of oxide paint, indicating very high reducing conditions in the bags. We also did some soil basic analysis to monitor nutrient availability before and after treatment. And then we rated disease on the plants. In the case of zinnia, we looked for the presence of a canker at the base of the stem. This is what Rhizoctonia solanae causes. And then in the case of Gerbera daisy, we looked at the presence of necrosis or plant death or cell death at the base of the crown, which is characteristic of Gerbera daisy, Phytophthora drexeleriae. So here are some results. As you see, when we used soil-less substrates, the treatment that had the carbon amendment achieved a high level of anaerobiosis compared to the anaerobic control. And in the case of soil-based substrate, which just as a reminder was a mix of comb-till and sand, in this case, we had a very high, even higher conditions of anaerobiosis, and it didn't really matter whether we had added the additional carbon source or not. Basically, the comb-till that was present already in the soil may have acted already as a carbon source, increasing the anaerobic conditions in the bags. When we looked at disease incidence, so this is the presence or absence of the canker on the plants, the wheat bran and tomato pomace treatment had much lower disease incidence compared to the rest of the treatment. So they were successful at controlling disease when the soil-less substrate was used. In the case of soil-based substrates, all treatments, including the anaerobic control, so the soil that didn't have the additional carbon source beyond the comb-till, they all reduced disease significantly compared to the control that did not receive anaerobic conditions. Beyond incidence, which again is the presence or absence of a canker, we also looked at the severity of disease. So how much disease was observed on this plant. And we did that by using a 1 to 4 scale, where 1 was a healthy plant, 2 was a plant that had a very small canker at the base, 3 had a large canker, and then 4 was a completely dead plant. And as you can see, we monitor disease weekly from 7 to 35 days just to observe the progression of disease over time. And within 7 days post-transplant, all carbon-amended treatments that had undergone anaerobic disinfestation maintain a very low level of disease all the way to complete healthy plants, versus the 2 controls had a high incidence, a high severity within 7 days post-transplant. And that trend remained throughout the experiment, except the soybean meal carbon amendment was not successful at maintaining plant health and reached high level of disease compared to the control. When we looked at the soil-based substrates, these work extremely well. Again, all anaerobic treatments, including the comb-till without the additional carbon source, had were significantly different from the aerobic control, and all plants remained healthy throughout the experiment. Because a picture is worth 1,000 numbers and 1,000 words, you can see here the difference between pots where the plants were planted in the control treatment, so where all plants died, and then plants that were planted in carbon-amended treatment after it underwent anaerobic disinfestation. So this was very reassuring and very interesting, except that when we looked at the effect on Phytophthora root rot, we did not observe the same results. As a matter of fact, this technique was absolutely not effective at controlling Phytophthora root rot. And in some cases, it appeared that the presence of the additional carbon source was enhancing disease incidence and severity as opposed to controlling it. So we were wondering whether the anaerobic conditions are actually a stimulating factor for Phytophthora root rot, or if the additional availability of a carbon source is also a stimulating factor. As we know, Phytophthora is an oomycete pathogen, and this pathogen thrives in wet environments. So the flooding of the soil may have just enhanced disease severity as opposed to suppressing it. However, we decided to look further into Phytophthora root rot to identify whether maybe lower amounts of inoculum in the soil or a different age of the plug of the plant that was planted in the treated soil could have an effect. And so we did a second experiment where we used 5 different rates of inoculum, from very little to the normal amount that we had used in all the other experiments. We selected wheat bran as our gold standard, and then we used plugs of Gerbera that were from between 2 and 10 weeks old to see if the age of the plant also had an effect. And we did find that the plant age, generally plants that were 6 or 8 weeks old, had lower disease severity compared to the rest. And that in the case of lower amount of inoculum, we had less disease severity. And however, there was no difference between ASD treatments, between aerobic and anaerobic treatments. So wheat bran actually in some cases showed higher disease incidence, again, reinforcing the fact that the availability of that carbon source may be stimulating for the plant. These, however, still gave us some ideas that maybe in conditions of not heavily infested soils, so more of a preventative measure, this technique could still be feasible for Phytophthora root rot. So in summary, this proof of concept study highlighted that ASD has very good potential for controlling Rhizostonia stem rot in both soilless media and soil-based media, and should be further evaluated in a variety of production conditions. We also found that suitability of this technique for controlling Phytophthora root rot may not be appropriate. However, it may still be effective when inoculum levels are low and different carbon sources are used as soil amendments. There's a lot of literature out there about ethanolic heavy carbon sources that can actually have an effect in suppressing Phytophthora. We did not test those specifically in this experiment because we had to make choices about what type of ingredients to use. So based on these results, this last year, we set up a second study. This was a study done outdoors. So we decided to move away from the completely controlled environment and moved into a field setting. And we set up a study with raised beds where my whole lab helped out with this. And we set up these raised beds to compare tomato pomace, wheat bran, and a non-amended control treatment within the same bed, so that we had a side-by-side comparison of the different treatment. And we incorporated the carbon source in the soil where we had artificially added Rhizostonia solanae. We then saturated the soil with water and then covered it with tarp for a period of 8 weeks. This was much higher in terms of timing than the 4 weeks that we tested in the greenhouse. And that was due to the fact that we had a very cold beginning to our summer, and we had to extend that incubation to make sure that the 25 Celsius temperature was maintained during the incubation period. At the end of the incubation period, we let the beds air out, and then we transplanted 2 different cultivars of zinnia into the beds and monitored these beds for disease development. And as you can see, we had a variety of plant responses, from some plant failure to complete plant failure, depending on the cultivar and depending on the treatment, which is briefly summarized here. We see that the control treatment, so again, this is the soil that did not have any carbon source incorporated, reached about a 45% disease severity based on cultivar, versus tomato pomace had a much lower disease severity, and then plants planted in the wheat bran treatment did not develop any disease symptoms throughout the experiment. So this is very reassuring. And so I want to conclude by saying that these are still very much work in progress. Daniela has graduated at the end of the summer, and a new grad student, Ambrosia, that you see pictured here, has just joined the lab, and she will continue the work with ASD. And particularly in the next 2 years, she will conduct studies in cut flower farms. We have identified industry collaborators that are interested in comparing ASD to soil steaming. And we will be doing these in soils that are naturally infested with pathogens. So not places where we actually put the pathogen, but may have a variety of different pathogens in them. We will look at different carbon sources, different amounts of carbon sources, to make sure that we identify specific amounts that are effective, but also cost effective for growers. And then we'll look at expanding the array of pathogens and amounts of inoculum for these different pathogens. So Ambrosia has a lot of work to do. So I would like to acknowledge all the funding sources that have contributed to this work, as well as all my lab members and collaborators that have contributed in many, many ways to conducting all this work. And that includes my grad students, my undergrad students, and as I said, faculty collaborators in the Department of Plant Pathology and Horticulture and Crop Science, and also Growers Talk and OHP for sponsoring this session today. And if you have enjoyed this session and you are interested in hearing more about cut flowers, here is information about next session on November 21. So if you're interested in registering, feel free to click on the link that is highlighted here. And at this time, I'll be happy to take your questions.
All righty, Dr. Hand, thank you for your presentation. Like she mentioned, we'll go ahead and open up the session for a question and answer right now. So I do have one question here in the chat asking about carbon source options. I know you touched on some. Could you talk about where people might be able to find them and what other options might be out there?
Sure. There's a lot of different options for carbon sources, and many different carbon sources have been tested over the years in different systems. One thing that we stress the importance of is the choice of carbon sources should be something that is easily decomposable. So we don't want to give something that then the microbes cannot break down easily. It should be a carbon source that is readily available locally. You don't want to ship it in from, far away. So usually byproducts of local agricultural commodities are good choices. And this explains why we picked, soybean meal and wheat bran and tomato pomace in Ohio specifically. But there's a lot of different options. Anything from molasses and ethanol, different brassica species that are already used as cover crops, tomato, wheat bran, wheat meats. There's really a lot of options.
Awesome, thank you. Mm-hmm. So when in the production cycle would ASD be a suitable option?
Yeah, so this is something that we have been trying to identify together with growers. It really depends on, the area of the country where the farm is located and what is their production schedule. We have had success here identifying the middle of summer as is appropriate time to do ASD. So right before the fall crops go in the ground, there's usually some times where there's some high tunnels that are empty. And so we are actually going to do that next summer with our industry collaborator, and we're going to have a side-by-side comparison with doing soil steaming and doing anaerobic soil disinfestation, and even biofumigation, so using cover crops that then get pulled into the ground and they decompose basically.
Awesome. Sounds like we'll have to have you back here next year after the results, right. Everything comes in. So I have a question here from Beth asking, does ASD still support a healthy microbiology diversity, or does it choke them out too, and then hope for them to return afterwards?
Yeah, this is a very good question. Thank you for asking it. So the ASD, this is one of the advantages of using ASD compared to a physical treatment such as soil steaming or solarization, where you have this heat that is reached that is very high, which kills everything that is in the soil, including the good stuff, right? So in the case of ASD, all that you're doing is basically providing a food source to the microbes that are present in the soil. And by shifting the environment to anaerobic, you just make a select group of organisms more competitive. And so they take over, and that taking over the environment in that specific moment, kills the pathogens that are strictly aerobic. So at the end of the system, you don't have that everything, the entire microbiome of the soil is dead, but only the aerobic organisms, which then over time, right, as you air out the soil, they will repopulate and at a much faster pace than with physical means such as solarization and steaming.
Awesome. That was a great answer.
Yeah.
So another question here. How do costs compare using ASD and other options?
So these very much depends on the type of carbon source that is picked. We have some data, locally in Ohio, for example, we can buy wheat meats at about 16 cents per pound. And so if you consider that our high end of application is like 9 tons per acre. So if you have a 30 by 96 high tunnel that is about, a little short at 1,200 pounds, which is about $200. If we have to, then of course you have to add the labor and the plastic, and that is all extra costs. But just for the carbon source, that's about the price. So I'm not 100% sure how that compares with what is currently being done, because it depends on, case by case, but that's approximately the cost for it.
Awesome. Thank you. And then I got another question here. How long does ASD typically showing like effectivity from the treatment?
Yeah, so that's another good question. And it depends on a lot of different things. So the type of soil that you have is going to make a difference, right? If it's a more compact soil, if it's a more porous soil, the amount of like how deep you incorporate the carbon source. We usually recommend anything between 6 and 8 inches of soil to be worked with that carbon source. But it's a technique that I would probably recommend doing yearly. It's uncertain right now how more often you would have to do steaming or other type of techniques. Considering that those also kill the good stuff, right? So you don't want to overdo things with those physical methods. But yearly or every other year would be what I would recommend in this case.
Awesome. So I have a question here from Shay, mentioning that they have very poor soil in Florida. Is ASD something that would work there?
Yeah, actually, one of the only studies that has been done in the United States was done in Florida, and the target in that case was nematodes, which is something that we see often in sandy soils, such as the Florida soils, and they had pretty good results for the control of the nematode. So I would definitely be recommending the technique in porous media.
Alrighty. Well, Dr. Hand, I'd like to thank you. That was a wonderful presentation. I learned a whole lot. I'm going to go ahead and wrap up here. I want to thank everyone else for joining us today.
We have a few questions waiting in Q&A.
All right. We've got some last minute questions sneak in here. Thank you, Karen. All right. So a couple more questions here. We have any thoughts on using ASD to address Fusarium wilt on Lisianthus?
So I'm going to get back to you on that, because one of our target for next year's studies is actually Fusarium wilt in Lisianthus. We have seen so much Fusarium wilt in Lisianthus that we've decided to select that as our next study system. So the other 2 studies that have looked at ASD, the one in Argentina and the one in China, Fusarium oxysporum was actually the target. And they both, it wasn't Lisianthus, the crop. I think in one case was snapdragon and the other one was carnation. But in both cases, they did see an effect of ASD on Fusarium wilt. So we are very much interested in looking at that ourselves, because Fusarium is very hard to control even with chemical means.
Alrighty. Here's another one. Do the carbon sources have an effect in the 2 pathogens you tested in vitro as well?
So I'm assuming that, so the Rhizoctonia and the Phytophthora, assuming we're referring to the 2 systems that we've explained. So the first set of slides that I presented, we're not in vitro per se, because we didn't conduct them in a petri dish, but we did use, plastic bags as our containers. And so it's very similar. I would consider that an in vitro experiment. And yes, they did have an effect on Rhizoctonia, but not on Phytophthora. That's when, we realized that with Phytophthora, we've got to look at possibly different carbon sources. I think that the carbon sources that we selected were maybe not the best for these pathogens. So we'll look further into that.
Alrighty, and I got another question here from Shay. have you guys seen any effect of pH on the treatment in any way?
Yeah, so we have not looked at the effect of pH pre-treatment, but the ASD treatment does impact pH and it does impact nutrient availability. So I did not report the results in this presentation, but we did see that it's important to conduct a soil analysis after the treatment, which is something that I would recommend, yearly anyways, to look at what type of nutrient content you have in the soil, what's available and what not, and adjust fertilization accordingly based on how the availability shifts post-treatment.
All righty. I think that wraps us all up. I think we're all good. Checking here in the chat one more time. All right.
All right.
All righty. Thank you again, Dr. Oh, did I get one more? I got to thank you.
All right.
from
Thank
Shay.
you.
So we're going to go ahead and start wrapping up. I'd like to thank everybody again for joining us today for another session of AFE's Grow Pro webinar series. And remind you again to join us next month for Dr. Roberto Lopez's presentation on cut flower production in the northern U.S. on Tuesday, November 21st at 1 p.m. Eastern Time. You can go ahead and pre-register at endowment.org/growpro. And while there, check out past webinar recordings, other grower-related resources, and research reports, all available to you for free, thanks to all the industry support that AFE has. We ask you to please complete the brief survey about today's session, and where you can suggest additional topics and help us to continue to improve these webinars in the future. I'd like to thank everybody again for joining us and wish everybody a great day.